It's really a pleasure to be here and participate in the Institute for Global Health, which has been created in a seminar series created over the last few years, and to be introduced by Miriam, and congratulations on becoming the head of the malaria part of it. In looking around the audience, there's a number of people here. I could spend the entire time talking anecdotally about my life with those people. So, but others, some of you may not appreciate it as much as them, and they may not even appreciate it. So I'm not going to do that. But what I do need to say is that I have had been associated with the university. Never was on the faculty here. Didn't go to school here. But I've been associated intensely with the University of Maryland since 1980. When I went to Indonesia, I joined the Navy in 1980. I had been at UC San Diego and on the faculty there, but wanted to become a real tropical medicine doctor. So I signed up to go to US NAMRU 2 in Jakarta, Indonesia. And during the first 3 months that I was there, I went on rounds in the infectious disease hospital of Jakarta, and I saw about 100 patients with typhoid fever, and 15% of them had died. So who in the audience can tell me to the person who developed the first antibiotic for typhoid fever? And not some of you old guys who actually know. Any of the younger people know who developed that and what his relationship was to the University of Maryland? So it was developed by Ted Woodward, who started all this stuff here, and went on to be the chairman of the Department of Medicine. So I saw all these patients. I wrote letters to 10 doctors, the best in the world, because there I was, this young physician out in Indonesia, and it's supposed to be that since Ted Woodward had discovered chloramphenicol for typhoid fever, the mortality rate for typhoid fever was less than 1%. And that's what I had learned at the London School of Hygiene and Tropical Medicine. And I was having a 15% mortality rate. And I described a syndrome of, for lack of a better word, cerebral typhoid fever, which was associated with a 55% mortality rate. I wrote to all of these experts around the world. They either wrote back, "Dear Dr. Hoffman, you're an idiot, you're a bad doctor," or didn't answer me, but Dr. Woodward wrote back and said, "Dear Dr. Hoffman, sounds like you're seeing some good old-fashioned typhoid fever, and I'd like to come to Jakarta to see it with you." And here was the most prominent guy in the entire field writing to me, this lowly little physician, and he actually came. I got stuck on a grass landing strip near Timor, and didn't meet him that time, but then I came back here in November of 1980, met Ted, met Shelley Greisman, who worked on endotoxin with him, and he convinced me to do, he said, I published a paper in 1951 that showed that hydrocortisone, steroids, reduced fever in typhoid fever, and since then, nobody really thinks that you should treat infectious diseases with steroids. And in fact, we had been taught emphatically in medical school and during residency that was the worst thing you could do. But why don't you do a study of that? And I said, Dr. Woodward, you're asking me to do a double-blind, placebo-controlled trial with death as the primary outcome variable by myself in Indonesia. I'm not sure I can handle that. And he said, not to worry, I'm going to send out one of my associate professors in infectious diseases. And then the associate professor became an assistant professor who became a infectious disease fellow who became the chief resident. None of them showed up, but his son, Bill Woodward, who was more interested in vaccines, and from there we actually did one of the major trials that led to licensure of TY21A, the oral typhoid vaccine, which was also pioneered here with Mike Levine's work in Chile in particular. But the first challenge study had actually been done here also at the University of Maryland. And so anyway, we ended up doing that study, and that is my most my most proud accomplishment of my life. All right, so you're going to hear about a lot of stuff that we're trying to do with malaria, but it was one of the first studies that I ever published, which was published in the New England Journal of Medicine in 1984, where we actually took that 55% mortality rate and reduced it to 10%. And showed and saved lives. And when I left Indonesia, there wasn't anybody dying of typhoid fever, but that was instigated from the University of Maryland by Ted Woodward. And since then, have had an enormous numbers of interactions, which I'll tell you about. So I'm going to be talking about malaria vaccines. And why am I working on malaria vaccines? Because I started out as a clinician working on tropical infectious diseases of many varieties. And had an amazing initial success with typhoid fever and with cholera and with lymphatic filariasis, but then turned to the most important tropical infectious disease, which is malaria. Certainly the most important tropical parasitic disease. And this is a child who is comatose with cerebral malaria in Indonesian West New Guinea, Jayapura, then called Irian Jaya, now called Papua. And in the last 100 years of research on treatment of cerebral malaria, with the exception of moving from quinine to artesunate, which reduces mortality, and despite, I don't know, tens of articles in the in the New England Journal of Medicine and the Lancet on treatments, potential ancillary treatments for cerebral malaria, nothing has reduced the mortality of cerebral malaria by 1 percentage point other than good primary medical care. And this was a child who I was called to see. We were doing a study, which I'll show you in a second the results of, in which we took the same high-dose dexamethasone that we used to reduce mortality in severe typhoid fever and did a double-blind placebo-controlled trial in cerebral malaria in Irian Jaya. And just before starting that trial, I was out there taking care of patients and doing a study, and I had admitted a young girl who was like 3 years old who had severe malaria, high level of parasitemia, but wasn't comatose. And at 2 in the morning, I got a call from the nurses, come to see this patient. Went to see her, and she was now cold, clammy, comatose with a blood pressure of about 60 or something. And we did a little dipstick, and I thought she would have hypoglycemia, and she had no detectable glucose. Gave her an injection of 50% dextrose, put her on a 10% dextrose drip, walked back to my room with my pediatrician colleague that Chris knows, Narain Punjabi, and we congratulated ourselves on the fact that if we hadn't been there, that girl would have died. And an hour later, I got a call from the nurses that girl died. This is the story of malaria. This was the trial, and the trial didn't work either. It had no impact on the mortality in cerebral malaria. And one of the things that I also did along the way was organize the first sequencing of the Plasmodium falciparum genome, an effort that was led by Claire Fraser, who's sitting here, getting together with the team at Tiger and Malcolm Gardner and so on, and the sequence of Anopheles gambiae, the major mosquito vector. And I had the honor of, there's something that we created called the Multilateral Initiative on Malaria, and in the meeting in 2002, I gave one of the keynote addresses on the Plasmodium genome with a follow-on on the Anopheles genome that we had also sequenced, and talked about the promise of systems biology, the promise of genomics, proteomics, transcriptomics for actually impacting malaria. And we're sitting here 15 years later, and we're still talking about the promise of systems biology, and we've had an amazing number of papers published, but as far as I can tell, systems biology hasn't eliminated one case of malaria. Not that it won't, not that we don't have much more to learn over time, but we have to do things. And so an enormous effort began about 10 to 15 years ago in which now over $3 billion is being invested annually in malaria control efforts, insecticide-impregnated bed nets, residual insecticide spraying, early diagnosis, treatment, intermittent treatments, and so on. And yet, depending upon who you read, the first number there, 438,000 deaths in 2015 comes from the World Health Organization, 666,000 comes from the Lancet, and 730,000 in 2015 comes from the New England Journal of Medicine. So there is, despite this amazing investment, there is still, 500,000 deaths from malaria annually. Now, one of the diseases that we often think about how impactful it was and how vaccines have impacted it is polio. So does anybody know at the peak of polio in the United States, how many cases of polio there were? The most number of cases, not deaths, but cases from polio. Anybody? All our infectious disease global health experts here? It's 53 to 57,000 in 1953 when there was mass hysteria in the country, and there were about 2,500 deaths. In the mid-1980s, the World Health Organization embarked upon a polio eradication campaign because of what a problem it was. And how many cases in the entire world were there of polio? Between 400 and 500,000 cases of polio in the world annually, and with death and paralysis occurring in something like 1 in 100. So malaria last year, in the face of $3 billion investment, had more deaths than polio ever had cases. Now that doesn't mean that polio isn't important, but it gives you an idea of the magnitude of the problem we are addressing in trying to fight malaria as compared to almost any other infectious disease. And there were also 214 million cases of malaria last year, according to WHO, which some people think is an underestimate, and that was no different than 2014 or 2013, despite this investment. So over the past decade, there's been a lot written about halving of the number of deaths, or at least from malaria, but there's a plateau that's occurring. There's no question about that the sort of low-hanging fruit, or whatever, for lack of a better way of putting it, has been met, and now we're seeing resistance, and we're seeing the measures that we have are not doing the same job that they did before, or they're certainly not reducing the cases or the deaths. So, as many of you will know, we've eliminated one infectious disease from the world, human infectious disease, which is smallpox, and that was done with a vaccine. Polio has been eliminated from the Western Hemisphere. That's been done with the vaccine, and we hope soon from the world. Measles has been dramatically impacted, and many other diseases. The most cost-effective, efficient way to impact any infectious diseases with a vaccine. So that's what I've been trying to do for the last 30 years. And so far, we haven't prevented too many cases. I'll tell you about more than anybody else has prevented, but not at the global level that we'll need to do that. So, but before going forward, I want to tell you one of the amazing parts of this process from a global health, from a global medicine type of perspective is how we've gotten here. So we've now raised or had invested about $270 million from the start of this project to take it over the finish line. The over $100 million of that comes from the NIH, mostly from competitive grants, but some from clinical trials that have been funded. We have the first investment by African countries ever, as far as we can tell, in clinical trials, certainly those above South Africa and in the Sub-Sahara. And we have $50 million invested by the government of Equatorial Guinea and 3 U.S. oil companies, with the majority of that coming from the government of Equatorial Guinea. We've gotten significant amounts from the Gates Foundation, the U.S. DoD, and so on down the line. And yesterday, our team from Tübingen met with the president of Gabon, with whom I met about a year and a half ago, and he promised the check was in the mail for the first $2 million of $9 million that he promised. We'll see. I'll keep you up to date on that. We have incredible numbers of collaborators around the world, of which Protein Potential LLC, our sister company, has been the major one, and the Naval Medical Research Center, but third on that list. And from there, from the beginning, has been the University of Maryland Global Health Institute and Genomic Institute, UMB, UNCVD. The names keep changing, but the characters don't. And I'm incredibly grateful to this phenomenal relationship that we've had. And we have something which is called the International PFS-PZ Consortium, which is about 200 investigators from about 40 organizations in about 20 countries who meet every 6 months to go over in real time the results of our studies. So we do adaptive trial design. We change in the middle of the road. We don't wait till we publish. We just had some papers, nice papers published, one by Kirsten Leik from here in PNAS, one in Nature, one in Lancet Infectious Diseases. This team knew about those results a year and a half ago. We don't wait to tell everybody what, to the day of publication. So this has been an extraordinary effort. All of these people come to this meeting on their own dime. We don't pay for it. And in most cases, they raise the money for their clinical trials because they're so excited about it. So this is another approach to funding global health initiatives, which we've been fortunate enough to be successful to some extent in. So we're trying to develop a vaccine, and we have two major focuses or foci. One, of course, the most important, the raison d'etre, is a malaria vaccine for use in regions endemic for malaria. And the other is a malaria vaccine for travelers from non-endemic areas to endemic areas. On the left side, we're not interested, not pursuing, at least now, and I would never say never, a vaccine to be used in the expanded program for immunization in a childhood vaccination program. And I can explain later why that is. What we're interested in is having a vaccine that we can immunize the entire population to halt transmission and eliminate the parasite. And this would, of course, have to be done with the other interventions that are being carried out right now. We also think that a vaccine to prevent, other than young children, pregnant women have the most morbidity and their offspring the most mortality from malaria. And so a vaccine that could be used to immunize adolescent girls and boosted at the time that they get married or the time in the first trimester is something we're exploring very vigorously. And then, of course, the other side is if any of us travel, or military, diplomats, Peace Corps, expatriate workers, to have a vaccine that could be used to prevent malaria. To be useful in either of those ends there, we think the vaccine is going to have to prevent P. falciparum malaria in 75, 80, some significantly high percentage of recipients. And just to give you a perspective on that, of course, you know that flu vaccine, which we get every year, some years protects 30%, 35%. Flu mist, which was not used this year, protected 3% last year. What's the largest selling vaccine in the history of the world from a money point of view? Anybody know? Prevnar, right? So when Prevnar went on the market as Prevnar 7, almost 20 years ago, 18 years ago, it had an efficacy of about 50 to 60%, and it didn't work in a lot of parts of the world because of the strains of streptococcal pneumoniae, and now it's been expanded to Prevnar 13, and you have Merck with a 22 antigen polysaccharide vaccine. So the precedent for most vaccines is they actually don't work that well, but we think that we're going to have a significant level of efficacy. So what occurs, this is the life cycle of Plasmodium falciparum, and you have a mosquito that injects sporozoites from dusk until dawn. The sporozoites are uninucleate, one nucleus. They're in the circulation probably for less than 5 minutes before they enter liver cells, where a uninucleate sporozoite develops to a mature liver stage schizont with 10,000 to 40,000 nuclei. So there is enormous amplification in the liver during the 5.5 days that it takes for the parasites to develop. When a liver schizont ruptures or the parasites are released, each one of those 10,000 parasites, called merozoites, can invade a different erythrocyte and during the course of 48 hours develop to a mature erythrocytic stage schizont, which then ruptures and then can invade another 20 red cells, 10 to 20. So one gets on average about a 10-fold replication every 48 hours in the bloodstream of the burden in the body of parasites. Alternatively, they can develop to gametocytes, which when taken up by mosquitoes, this is the sexual stage, develop during a course of about 2 weeks to sporozoites again. There is no pathology, symptoms, or signs associated with the sporozoites in the circulation, the parasites developing within the liver, or the sexual stage here. It's only the cycle of invasion, development, rupture, and reinvasion that causes the disease we know as malaria. So if one were to prevent the parasites from ever getting out of the liver into the bloodstream, one would prevent all disease and transmission. And that's the goal of our program. The sporozoites, depending upon which ones we use, and I'll tell you about that, but the sort of classical one is radiation attenuated sporozoites. They go into the liver, partially develop, and stop. They're metabolically active but non-replicating. Now, all of the licensed vaccines that we have, which are attenuated vaccines, are either replication deficient or non-pathogenic. They're not non-replicating. So this is a bit different. You don't get more of irradiated sporozoites than what you put in. And the two ways that you could approach this would be preventing the sporozoites from ever getting into the liver, and these are extracellular, so there's only one immune response that we know of that can work in that period of time against those sporozoites. Anybody? Antibodies. It says so on the slide, so that's pretty good. But they have to work. If 100 sporozoites are injected, 99 are knocked out. In a non-immune individual, a week later, there's 10,000 to 40,000 parasites emerging that then replicate tenfold every day. The other approach is to attack them when they're in the liver, when you have 5.5 days without symptom, signs, or pathology, and that's by T cells. And primarily, we believe, CD8 T cells. So these are the sporozoites here. They invade hepatocytes. During the first 3 days, they round up, and you can't distinguish an irradiated versus a non-irradiated parasite at 3 days. But at the end of 3 days, the irradiated sporozoites don't replicate, and the new ones replicate. The non-irradiated replicate 10,000 to 40,000 times. So the immune responses that we generate with irradiated sporozoites have to be against either the sporozoites or these early liver stages, of which there's about 1,000 proteins expressed. So it's against many, many different proteins. So our platform technology at Syneria that we had to figure out how to do was to make aseptic, purified, vialed, cryopreserved Plasmodium falciparum sporozoites that meet all regulatory standards. And our first product was what we call PFS-PZ vaccine, which are radiation attenuated sporozoites. The second was PFS-PZ challenge, which is live sporozoites, which are used to actually infect people. The third is PFS-PZ CVAC, which is giving live sporozoites with an anti-malarial drug. And then the final one, which is going to start clinical trials, which will be the world's first genetically attenuated sporozoite vaccine, is called GA1. And they are genetically attenuated by knocking out 2 genes, and they basically develop more or less like irradiated sporozoites. So we have done 32 clinical trials of our PFS-PZ based products, and at least if I'm not missing anything here, 9 of those have been done in collaboration with the University of Maryland, which I think is in first place. I didn't count everybody else, but I'm pretty sure there's nobody very close, all right? So we did the first PFS-PZ challenge mosquitoes. So before we got aseptic sporozoites out of mosquitoes and cryopreserved them, we wanted to make sure the sporozoites still worked. So in two trials, one with Kirsten Leik as the lead, and the second with Matt Lorenz in the lead, and Kirsten is the senior author, we subjected people to the bite of these aseptic infected mosquitoes and showed that they were more effective actually than the traditional mosquitoes. We then did the first clinical trial of PFS-PZ vaccine. It was a collaboration with the Naval Medical Research Center, published in Science in 2013. Another trial with second trial at the Vaccine Research Center at the NIH, published in Nature Medicine in 2016. Another one, sub-element of that Kirsten published in PNAS in 2017. A trial has just started in Burkina Faso with Matt Lorenz as the PI from Maryland. We've done PFS-PZ challenge, which is the live sporozoites, which I ran out of time putting in the references published by Kirsten Leik, and 7G8, a second strain done by Matt Lorenz early last year. And now we're doing the first trial in Africa with natural exposure, looking at naturally acquired natural protection against natural infection. We'll start next week in Mali with, again, Matt and Mohamedou Thera as the PI. So we at Scenaria are incredibly grateful for this collaboration. There would be no Scenaria have gotten to the point it is without the University of Maryland, so thank you. And just to close put another bookend on the University of Maryland, my second son, who is graduating from medical school, is starting an internal medicine residency here next month, in June. So I will finally have a tangible, physical relationship with the University of Maryland, or family relationship. So we have a product pipeline. We have different vaccines at different levels of the process. And I'm not going to belabor this and just go through it, but we expect that within 6 months we'll have a finalized 3-dose regimen that meets our requirements for non-immune adult travelers and military. And we then intend to conduct phase 3 clinical trials and submit a biologics application, license application to the FDA in the US, and follow within a year in the European Medicines Agency and all age groups in Africa. So what do you need to license a vaccine? It has to be safe, it has to be well tolerated, and it has to be efficacious. But it actually doesn't have to be that safe. We've got vaccines that have significant side effects. We have lots of vaccines that hurt and cause even febrile seizures, which is the GSK RTSS vaccine had an increased incidence of a number of adverse events, including febrile convulsions. And we know that a lot of vaccines don't work that well. But we think that we have to be extremely safe, well tolerated, and efficacious. And we've now done multiple double-blind, placebo-controlled trials where our comparator is normal saline, not another vaccine that has side effects. And in those trials, we have had no adverse events. There's no difference between giving salt water and our vaccine, which is a tribute to the manufacturing team. And in a number of trials in Africa, 97% of the recipients said they didn't have any pain associated with the injection. So that's pretty good from a development point of view. We've been very fortunate to have Alan Cross be on I don't know how many safety monitoring committees and data safety and monitoring boards, but really critically important to the development of our vaccines. So what about efficacy? So we've developed sort of a framework for how to get better and better efficacy, and it's shown here. This was published in a paper by Judy Epstein earlier this year in JCI Insight. If we start with minimal or no protection, and then we want to go to short term, which we've defined for now as 3 weeks, and that's pretty much the standard in the malaria field, 2 to 4 weeks, to long term, which we've been calling 5 or 6 months, and go from what we call homologous protection, where you immunize with parasites and challenge that are the same strain, so they have the same sequence, to heterologous, where the immunizing parasite and the challenging parasite are of different strain, different sequence. So we have short-term homologous, long-term, short-term heterologous, long-term homologous, long-term heterologous is what we need. This is all with controlled human malaria infection. In the field, of course, it's always going to be heterologous or heterogeneous protection. And we identified that to go from this to this over multiple thresholds, you're going to have to vary the number of sporozoites that you inject, the number of injections, the spacing between those injections, and perhaps the composition, even by adding in heterologous strains of parasites. So the first milestone to achieve was short-term homologous protection. And this is a paper published in 2011 in Science. It's the first research article ever published in Science on a clinical trial of a malaria vaccine, with Judy and Kirsten as being co-first authors. And it was given subcutaneously or intradermally, and it was safe. It was well tolerated. The immunogenicity was terrible. And in the best group, we got 2 out of 16 people protected. So that's a far cry from my 80% protection. So how did we ever get this published in Science? And the answer was that in collaboration with Bob Cedar at the Vaccine Research Center at the NIH, we took the exact same vaccine at the highest human dose, and we gave it to rhesus macaque monkeys, non-human primates, either subcutaneously, as we had done in that trial, or intravenously. And if you remember, we believe that T cells attacking hepatocytes are the mechanism of protection. We waited 4 months, took out the livers, isolated the T cells, established that in the animals that got the vaccine subcutaneously, as in humans, there were no sporozoite-specific CD8 T cells in the liver producing interferon gamma, which is what we think is important. And in those who got it IV, 3% of all the CD8 T cells in the liver were specific for sporozoites and produced interferon gamma. So this implied that we could get what we want. That's kind of the holy grail of pre-erythrocytic stage malaria vaccine development, but route was really important. I won't go into all the stories of what it took to getting this IV, but 2 years later we published a study with Bob Cedar as the PI showing that we could get protection. And what we did was we gave 5 doses of 135,000 sporozoites, a total of 675,000 sporozoites, and we got 100% protection, 6 out of 6 at 3 weeks. So we achieved short-term homologous protection. In the entire world's history of clinical trials of malaria vaccine, of any vaccine, there had been only 1 or most 2 studies in where anyone had ever gotten 100% protection in a vaccine trial of anything, certainly not malaria. But then we went ahead and we took 6 of those protected individuals from 2 groups, challenged them at 20 weeks, 5 months, looking for long-term homologous protection, and we only got 33%. So we weren't good enough yet. So then, according to this, the next thing to do is increase the dose. So we went up to 5 doses of 270,000 sporozoites, a total of 1.35 million, doubling the total number. And this is a paper that was published in JCI Insight in January, and we repeated the short-term homologous protection, 12 out of 13 at 3 weeks. We got short-term heterologous protection, 80%, 4 out of 5 at 3 weeks. We didn't quite meet our 80% long-term homologous protection, but we got 65%. If you multiply, we took 10 of these protected people, and 7 out of the 10 were protected at 24 weeks. 7 out of 10 times 12 out of 13 becomes 65%. We also showed that you could go from a 5-dose to a 3-dose regimen to make it more practical for both short and long-term homologous protection. But when Judy challenged with 7G8 at 24 weeks, only 10%, 1 out of 10 were protected. 4 out of 5 times 1 out of 10 is 8%. So we didn't achieve long-term heterologous protection. So what should we do? Increase the dose, right? But first, we actually wanted to look further at long-term homologous. So the team at NIH with Kirsten took individuals and only challenged them first at 5 months. and got 6 out of 11 or 55% protection. Then took the 6 protected people, challenged them at 14 months, and they were all protected. So it looks like you can sustain the long-term homologous protection. We still weren't at the level we wanted to be at, but way better than anyone else had ever been. We then went to Tanzania with the exact same regimen, 5 doses of 270,000, and did look for where we'd gotten 12 out of 13 with short-term homologous protection, and lo and behold, we only got 20%. 4 out of 20 were protected, and those 4 were also all protected at 24 weeks. So taking the exact same vaccine with the exact same regimen and doing the exact same type of challenge, we got far fewer people protected, or a lower protection rate in Tanzania. And I'll get into in a moment, but they had 6.5 times lower levels of antibodies, much lower T-cell responses also. So there was something about being in the field, that having natural exposure to malaria, that reduced things. And then we took that exact same regimen and went to Mali, working with the LMIV at the NIH and the team at University of Bamako Malaria Research and Training Center, with Mohamedou Sissoko and Sarah Healy as the PIs, and looked at protection for 6 months against natural transmission of malaria. And so what we had here was there were 44 controls and 42 immunized that made it through the study on a protocol basis. The attack rate of malaria in the controls was 93%. Now, one of the reasons why there's so much malaria is there's so much transmitted. There isn't any infectious disease that anybody can bring up here where you clear a person of that infectious disease, which is what we did. And over the course of 6 months, 93% of the adults with lifelong exposure to malaria became reinfected with the parasite, most of them actually within 10 weeks. And we followed them up for 24 weeks, and what we got was protective efficacy. The way that it's been reported in the malaria field with GSK has been by time to event analysis, which you take into account you get the infection and how long it takes you to get the infection, and that was 52%. That's the highest level of protective efficacy against infection ever seen with a malaria vaccine the first time we tried it. And if you look at proportional analysis, which is the way many of us think about vaccine efficacy, which means at the end of the 6 months, how many got infected in the placebo versus the immunized, it was 28.5%, both highly significant with confidence intervals that didn't come close to 0. Now, the GSK vaccine has been tested only once in that type of setting. And in that type of setting, it was in Western Kenya. They did it for only 16 weeks. The attack rate in the controls was 49%, and there was no significant protection. So the closest, that's the only comparator we have to any other vaccine that's been tested in this kind of setting. So what we found was that 5 doses of 270,000 sporozoites protection was at least as good or maybe better against intense exposure to heterogeneous parasites in Mali than against heterologous CHMI in the United States. Because remember, at 6 months in the United States, we only got 8% protection, and here we were getting 28.5% proportional analysis protection. And it was actually as good, if not better, than homologous short-term CHMI in Tanzania. So if CHMI controlled human malaria infection may be more stringent than intense natural exposure in the field. So what should we do next? We went up on the dose. And we went to 3 doses of 900,000 sporozoites. So we cut down on the number of doses, which we know is not quite as good. 3 is not quite as good as 5. But then we went up in the total doses to 2.7 million sporozoites, 3 doses of 900,000. And this is not published. In Tübingen in Germany, they've done 3 doses at day 0, 7, and 28. The other studies I showed you were at 8-week intervals, or 16 to 20-week immunization regimens. Here they got it all over within 1 month, and got 100% short-term homologous protection. with this contracted immunization regimen. Then they went to 2 doses at 0 and 7 days and challenged at 3 weeks and got 4 out of 6 protected. Now, in the entire history of the malaria vaccine development, all of the controlled human malaria infection studies that have ever been done for any malaria vaccine, the only one that's ever gotten more than 28% protection was RTSS, which averaged about 30 to 50, but has had 2 trials where they got more than 50% and more than 67%, but none of the others have ever gotten more than 28%. And here we are with 2 doses at 7-day intervals giving 67% protection. So this is, again, they've now doubled this dose, and we'll have data in a few weeks on a 2-dose regimen potentially. And then we had another paper, which was published with Kirsten in PNAS about a month ago, in which challenge was done long-term homologous at 19 weeks and got 64% protection. But when 6 of those protected 9 people here were re-challenged with heterologous parasites at 33 weeks, 8 months, 5 out of the 6 were protected. And again, 5 out of 6 times 9 out of 14 is 54%. So we've now achieved more than 50% long-term heterologous protection, which we had already seen long-term heterogeneous protection in Mali. So, and then we took that regimen and went back to Tanzania, where we had gotten 20% protection, and we got 100% protection. Short-term homologous, and now long-term homologous, we didn't achieve anything better. So we're still not quite there in the Africans. So we have a whole series of studies that have been published, and we now have many other studies that are going on in Tanzania, Mali, Burkina Faso, the United States. The Burkina Faso team here has now gone to 3 doses of 2.7 million sporozoites. In Kenya, we've immunized, I'll tell you about in a second, infants with up to 1.8 million, and so on. So trying to close in on finalizing a regimen that could be taken to phase 3. So the next thing we had to do was to immunize infants. So we went down in Tanzania from adults to adolescents to older children to younger children, and then infants, and showed that that was safe, well tolerated, and that the infants made much better immune responses than did the adults. And then we started a double-blind placebo-controlled trial in Kenya, in which we will be immunizing 416 5 to 11-month-olds. It began in January of this year, and thus far we've immunized about 350 infants have been immunized successfully there, and we'll know by the end of the year or beginning of this year how that worked. It's 450,000, 900,000, or 1.8 million, 3 doses at 8-week intervals with a 24 and then 52 and then 2-year follow-up depending upon the results. And then we've also started in Equatorial Guinea in 6-month-olds to 65-year-olds with the hope that we finally have a regimen that we can take to immunize the entire population. This is actually a year ago in Tanzania. These are the first 1 to 5-year-olds lining up to be immunized. And then here is the first infants. We were told over and over and over again we would never be able to give this vaccine to infants. We've now given it to 450 infants in 3 different countries, and it's been incredibly well tolerated. And here is, we give the vaccine by what's called direct venous inoculation. It's an injection of 500 microliters in about 2 seconds through a 25-gauge needle. And this, of course, babies cry when you get them ready to be immunized or injected or blood drawn. This particular child didn't, because it can't feel the needle going in. But the majority of them do cry. And so this is just one bit, and I knew Marcelo was coming here, so I figured I had to show one immunology slide. I've written many, many papers on immunology, but we're all about protection as the outcome, not immunology. But what I told you that in the adults, this is Tanzania, but also in Mali, we were seeing much, much lower antibody responses after a full immunization regimen than in the United States. And so this is the study that Kirsten published on, in which she had 9 protected and 6 not protected, or 5 not protected, VRC314. And these are antibodies 2 weeks after the last of 3 doses of 900,000 sporozoites against the circumsporozoite protein. And you can see here it's the average is about 12,000 titer. It's OD 1.0, a little bit higher in those protected than not protected, but it's not statistically significant. Now you go to the adults in Tanzania who also had 100% protection at short-term homologous, and it's 10 times lower. Now you go from the adults to the adolescents to the older children, younger children, and now the infants here have antibody titers as high as the adults in the United States and 10 times higher than the adults in Tanzania. So we think that there is a immune regulation, dysregulation due to long-term exposure to malaria that's going on that we need to solve. But we're very hopeful that the infants are really going to be highly protected because we don't have to solve it in them because they haven't been exposed before. So the summary of completed clinical data for PFSPZ vaccine, we've given it to greater than 1,100 volunteers, safely received greater than, I think it's now 3,000 direct venous inoculation injections of vaccine. Most who can be asked say they can't feel the injection. There's no difference in adverse events between placebo and vaccine. And of course, when we started, we had to make sure they didn't get malaria. So there's been no breakthrough infections after a total of over 2 billion sporozoites being injected. In terms of short-term homologous protection, we've had greater than 90% protection in 5 different clinical trials in the United States, Germany, Tanzania, and Mali at 3 weeks after the last dose. And we've had 4 trials with 100% protection. And as I told you, in the history of the world, there's only been 2 others that we know of that have had 100% protection. And certainly, we've done it with a 3-dose regimen in 3 cases and a 5-dose regimen in 2 cases. No other vaccine has ever achieved this even once, malaria vaccine. Short-term heterologous protection, we have 80% at 3 weeks. That's only 1 study. No other vaccine has ever achieved it even once. Today, Kirsten is not here, right, Kirsten? Oh, she is here. Kirsten is immunizing, boosting people, and will be challenging in 3 weeks about 25 people. So we're going to have much more short-term heterologous as well as long-term. It's a 6-month challenge in some of the individuals in 4 weeks, no, 7 weeks. So then in terms of long-term homologous protection, we've had 55 to 65% at 5, 6, and 14 months. We haven't yet achieved the 80, but we've not assessed our best regimens yet for the long-term homologous. And this has been done with 3, 4, and 5-dose regimens. In terms of long and medium-term heterologous, no vaccine has ever achieved it once, and we've had 54% protection against the non-vaccine strain in the United States at 8 months after the last dose. There was an intervening homologous challenge at 19 weeks, which may have impacted the longer-term heterologous protection. That was done with a 3-dose regimen, and then we have the results, and we've duplicated those with a 3-dose regimen in Mali. So we've done it with both a 5 and a 3-dose regimen in Mali. This just shows you the number of individuals. 1,391 subjects have received 3,365 DVI injections, ranging from adults to adults with the most, but infants closing in terms of numbers. So our research objectives in the set of trials that I've told you about have been to reproduce the findings, show that we could go to higher doses and continue to have safety and tolerability, look at durability, protection against heterologous parasites, reduce the number of doses, look at the feasibility of this DVI, and then get protection in Africans. And so we're moving forward, we're converting, we're shutting down our manufacturing, so by the end of the year we'll be at phase 3, licensure and commercialization compliance in terms of making the vaccine and planning phase 3 trials. We got fast track designation by the FDA last September, which will allow us to get over the finish line faster. But I want to close on, that's all about PFSPZ vaccine, but our best vaccine is what we call PFSPZ CVAC. And we published a paper in Nature with a team in Germany on this in February. And here we had 3 groups, 3 doses of 3,200 sporozoites, not 2.7 million, 3,200, a total of 10,000, 3 doses of 12,800, a total of about 40,000, and 3 doses of 50,000, a total of 153,000. And here we didn't challenge at 3 weeks, like I told you, we challenged at 10 weeks. And we went, it was like a mouse experiment, 33%, 67%, 100% protection. So 100% protection against controlled human malaria infection at 10 weeks. And then we truncated the regimen down to 10 days. So you immunize on days 0, 5, and 10, and got 5 out of 8 protected. And so that's now being tested in a number of places at a higher dose. What's going on? Why is this so much better with a 10th or a 20th of the number of sporozoites? So this is an hepatic sinusoid. These are the hepatocytes, endothelial cells, Kupffer cells. The sporozoites come in, and in the case of the radiation attenuated vaccine or genetically attenuated, which I'll tell you about in a second, they stop right here. They are metabolically active but non-replicating. They round up, but they don't produce more nuclei. In the case of the chemoprophylaxis vaccine, CVAC, they replicate fully, they release into the bloodstream, and then they're killed by chloroquine. So they replicate 10,000 to 40,000 times, and they produce 3,000 proteins that weren't there in the irradiated sporozoites. So we think it's a question of dose and breadth. And so we published one study when we gave it ID and it didn't work. This study I've just told you about, there's now been a study done at the NIH which duplicated these results and is looking at another drug, pyrimethamine. And there are now studies going on in Germany, Equatorial Guinea, the United States, in Seattle, in Mali, as I told you, is going to start next week, Ghana, Gabon. We have a huge trial that's going to start next year in Indonesian soldiers who are in Java and then go to Indonesia and West New Guinea for a 6-month deployment. And then in Kenya and at the clinical center at the NIH. So it's moving quickly. And here we have a whole nother concept which we think can revolutionize malaria control and elimination. And the idea is the following. Right now, in multiple spots around the world, people are going in with mass drug administration to reduce the reservoir of parasites. So if we were to use the mass drug administration drug along with our PFSPZ challenge and turn that into CVAC, which is chemoprophylaxis vaccine, one would eliminate the reservoir and prevent reinfection all in one fell swoop. And there's enormous enthusiasm now being generated to do this, to truncate this forever process that we're on of trying to control and eliminate malaria. So just a few innovations for 2nd and 3rd generation vaccines. We've now managed to figure out how to eliminate the mosquitoes from the production of sporozoites, and we're 2- or 3-fold more efficient in vitro than we are in mosquitoes. That will come in a few years, and that will replace mosquitoes. This just shows you that in a liver assay, at the end of 6 days, the mature liver stage schizonts that we produce are identical whether they come from mosquito salivary gland produced sporozoites or in vitro produced sporozoites, and the numbers are, if anything, greater with the in vitro produced sporozoites than the salivary gland. And then finally, we'll be starting at the end of May a trial of the world's first genetically attenuated PFSPZ vaccine with the team in the Netherlands in Leiden. And that's based on a double knockout parasite. And we're now producing Plasmodium vivax that we'll be able to vial and use for injections. We're looking forward to phase 3 in non-immunes. We are under no illusions regarding how difficult a task we have set for ourselves. We're trying to do something that tens of thousands of scientists working with billions of dollars over the last century, the first paper on malaria vaccines was published in 1912, have failed at, including the entire team here. So this is not simple. There are no human vaccines against parasitic infections. There's no human vaccines that are eukaryotic cell whole vaccines that are licensed. There's no vaccines that are delivered in liquid nitrogen vapor phase, and there's no vaccines that are administered for preventive vaccines by direct venous inoculation. These have all been, in the past we used to call those problems, then challenges, now we call those opportunities, right? But our motto is good is not good enough, and as an old Navy guy, I always go back to the first officer of the U.S. Navy. Anybody know who that was? John Paul Jones. And this is actually in all of our passports, this quote. It seems to be a law of nature, inflexible and inexorable, that those who will not risk cannot win. John Paul Jones. Thank you. This is the team that goes to work every single day with the dream and the idea that they're developing a vaccine that can save millions of lives.
Thanks so much, Steve. We have a very busy schedule for Steve, so if there's burning questions, we can have one or two.
Steve, this is fantastic. If you would have to put your eggs in just one basket, will you call CVAC the future?
So I don't know if everybody heard that. If I have to put my eggs in one basket, and I started out with multiple baskets and it keeps expanding, but you only have so much time in your life, and there's only so much money, and there's children dying every single day. The answer is, I showed you three things for a vaccine. It has to be safe, well-tolerated, and efficacious. There is no doubt in my mind that CVAC is the most efficacious. We can produce in the same space in the same time at 1/10 of the cost, 10 times to 20 times as much vaccine, but we're talking about giving live parasites that can kill you to people if they don't take the drug properly. So I believe that we can overcome that. And as it relates to elimination campaigns, mass vaccine administration, I believe that CVAC is definitely in the short term the way to go. You won't be able to give CVAC to pregnant women, in the short run at least, until you have a lot of data. So no matter what campaign we do, we have to consider irradiated or genetically attenuated sporozoites because they're so safe. I mean, it's unbelievable how safe they are. And then the other thing that we're doing, which I really didn't get a chance to go into, is that just like Prevnar going from 7 to 13, with enormous help that comes here from the Institute for Genomics, led by Joanna, and working with Claire, we are sequencing all the parasites and coming up with strategies for a multivalent vaccine, which may be the primary one, the first generation, but more likely is to be a second generation vaccine. Claire.
Steve, do you foresee one vaccine serving both purposes that you talked about in the beginning, for use in areas where malaria is endemic and for travelers, or do you see, based on your data so far, two vaccines?
So we've always envisioned one. We like to be simple, but actually malaria is very complex, and there's different settings that could, it's the interesting thing, just as an aside, I went to the WHO because we have to deal with them in terms of pre-qualification and all this stuff, and I walked in there and said we have 4 vaccines, and they practically laughed me out the door because nobody comes in with saying we're going to solve a problem with 4 different vaccines. But the fact of the matter is, it's the exact same manufacturing process. That's not 4 different vaccines. That's just 4 different sporozoites that are slightly, attenuated in a slightly different way. In one of them, it's radiation attenuated, and the other one, we don't do any attenuation. The other one is the same, type of cell bank. So I can conceive, we have a problem here that is huge, and everybody wants to solve it simply, right? And different groups, we have to have a vaccine that's practical and costs 4 cents, and they have all these target product profiles that are like this long and envision every single aspect. Our target product profile is safe, tolerated, and efficacious. And we believe that if we can get to that, there's tens of thousands of smart people in this world who will figure out how to get it to the people who need it at the right way, and it may take multiple products. I just don't know.
Thank you very much.
Thank you.